Alright, this is the first of a 4-part lecture. It's lecture 5, part 3. It's the finishing work or coverage of the topic of metabolism. Mostly we're going to focus on catabolism, and all of this information is in chapter 4 of your textbook. The goals for this part of the lecture are to review the role of and identity of electron-carrying molecules that are important in the cells, in all cells, including microbial cells, and to review how ATP is made. You might feel like we've been talking about that a lot so far, but really what we've been talking about is the topic of redox reactions and bioenergetics. So we've considered redox reactions that can yield enough energy that can then be conserved to make ATP, but we haven't really talked about the 3 biochemical methods whereby ATP is actually directly produced in the cell once you have access to that energy. Redox reactions in cells, the ones that we considered in class, those exothermic reactions that are energy yielding and wherein that energy can be harnessed or conserved to then make ATP, if they all happened in one single step, they would release so much energy that it would be harmful to the cell. So instead, those reactions are broken out into small incremental steps. Most of those are actually smaller redox reactions, and this generates a need to have electron-carrying molecules in the cell to link those reactions. Also, many of those reactions will in fact take place at different cellular locations, and that also generates a need to have, in some cases, diffusible electron-carrying molecules. So cells have 2 types of electron-carrying molecules. They have a type that is referred to as a prosthetic group and a type that is referred to as a coenzyme. Prosthetic groups are attached to enzymes typically. And examples of prosthetic groups are Flavin mononucleotide, FMN, and Flavin adenine dinucleotide, or FAD. Okay? These are common groups that are attached to proteins, which then earn the name Flavin proteins, and they're very important, for example, in electron transport chains. The other class of electron carrier are the coenzymes. These are diffusible, so these are molecules that are not typically found permanently attached to proteins or other cell structures. And examples there are NAD+ and NADP. So NAD stands for nicotinamide, adenine dinucleotide, NADP, its cousin, nicotinamide adenine dinucleotide phosphate. Shown here are the oxidized forms of both of those coenzymes. So FMN, in both its oxidized form on the left and its reduced form on the right. I know it's reduced quite simply by looking at the fact that it has 2 hydrogens associated with it, which therefore will provide electrons, and gaining electrons is reduction, is shown in this figure. So here on the left we see the oxidized form, on the right the reduced, and if we look at the center we see what can happen here. Okay, so when FMN gains 2 hydrogens and the electrons therein, it gains them at this position and this position, which reduces it to FMNH2. So what we see is in the electron transport chain, which we're leading up to discussing, we see protein complexes, and in complex 1 of the electron transport chain, which is what receives NADH during aerobic or anaerobic respiration, FMN is associated with flavoprotein in that complex. FAD, which is not shown in this figure but is a very similar molecule, the FAD, which is a prosthetic component of flavoproteins, is found in complex 2 of electron transport chains. One important feature of the flavoproteins is that they can accept protons and electrons but they only donate or transfer electrons. So shown here is the chemical structure for nicotinamide adenine dinucleotide. On the left is the oxidized form, on the right is the reduced form. Also, this is the structure for NADP. Here's where the phosphate group would go. Okay. So this oxidized form has a positive charge at this nitrogen, okay, and it is neutral over on the right-hand side in the reduced form. These are based on nucleotide structures. There is ribose sugar there and there. This is essentially a nitrogenous base form up here, and here's the phosphate group here and here. So you know a little bit about the chemistry of these structures. Now, these compounds are extremely important in cellular processes, NAD particularly in catabolic processes, NADP a little more so in anabolic processes, and they are electron carriers. They're diffusible, they are coenzymes, and what they're capable of doing is carrying 2 electrons and 2 protons, as I've written down here on the left. Okay, and the reaction is actually NAD plus 2 hydrogens. Okay, this is the reduction reaction, will become NADH plus a proton. Okay, so the NAD itself gains 2 electrons and 1 hydrogen, and the other proton is liberated into the environment, and that's actually really important. That is one of the sources of protons that are used to generate the proton motive force. So this is a really important point that I'm making, and it's important that you get this down in your notes for quiz and exam purposes. That is providing some of the protons important in the proton motive force. The rest of the protons are provided by the dissociation of water, but the reduction of NAD plus and NADPH in some systems is what yields some free protons in the environment that are then useful in creating the proton motive force. Let's look at this figure from your text that shows how NAD can facilitate redox reactions and makes the point that the NAD is not itself consumed, it is in fact recycled in these cellular processes. Let's start here on the figure. Here we've got the oxidized form of NAD, NAD+. We've got some electron donating molecule or substrate. Okay, so both of these are binding to enzyme 1. All right, so the substrate binds to the active site, but there's also an NAD+ binding site. They form the enzyme substrate complex, and as a result, the NAD+ is reduced to form NADH. Okay, and the donor has been oxidized. It has given up its electrons. Okay, that NADH is diffusible, and that's what these arrows are the point that they're trying to make. The NADH can then diffuse to another cellular location, to another enzyme complex, and this would be enzyme 2 down here. The enzyme 2 has an NADH binding site and an active site, and the electron acceptor is binding to that active site. This reaction then occurs because the enzyme has brought the NADH and this electron accepting molecule together in space and time into a transition state form, and what will happen then is the electrons associated with that hydrogen are being transmitted to the electron acceptor. So we've got electron transfer there, okay, and the electron acceptor is then reduced. It can go off. It's a product of this reaction, and NAD+ is regenerated and is then able to continue its role of receiving electrons in the cell. This figure is not making any points about protons as I did in the previous slide. It's simply making the point that the NAD+ plus is regenerated, okay? And in fact, you could think the NADH is regenerated too, that this is a cycling event, that these electron-carrying molecules serve to facilitate or connect disparate redox reactions that need to be linked in the cell. The point being here that the NAD role is to connect or allow the electrons to flow from this donor to this acceptor, even though they themselves are never actually interacting. I want to make one additional point here, and it's about enzyme 2 and its role. Enzyme 2 is serving to oxidize or catalyzing the oxidation of NADH. And so enzyme 2 in this figure is actually playing the role of an enzyme more generally referred to as NADH dehydrogenase, which is a very important enzyme in complex 1 of the electron transport chain that we're going to look at in a later part of this lecture. You should now know how cells harness the energy released in spontaneous and energy-yielding redox reactions to make ATP. From a biochemical perspective, there are 3 ways to make ATP: substrate-level phosphorylation, oxidative phosphorylation, and photophosphorylation. This last, of course, only occurs in phototrophs. Okay. The first 2 occur in respiratory organisms, substrate-level phosphorylation, if it's the only one used, is the only method for what are called strictly fermentative microbes. So just to rehash, 1 and 2 can both be used and are both used by respiratory organisms. 1 is the only way that strictly fermentative microbes make ATP, and for 3, the phototrophs are what do photophosphorylation. Substrate-level phosphorylation is the synthesis of ATP by transfer of a phosphate group from a nearby organic molecule directly to ADP. Another way of saying that is the transfer of a phosphate group from a nearby substrate directly to ADP. It does not utilize ATP synthase or the proton motive force. Okay, so ATP synthase or the proton motive force, not used in substrate level phosphorylation. Instead, we have an enzyme, it binds to ADP, and it binds to phosphorylated substrate. What that is going to be specific according to whatever enzyme is catalyzing this process. That enzyme then transfers this phosphate group from that substrate onto ADP, yielding ATP and a dephosphorylated product. Substrate level phosphorylation is important in fermentative processes, and it is also part of the glycolysis, which is a fermentation that occurs in respiratory organisms. In the case of these organisms, those that are gaining their ATP entirely through substrate level phosphorylation, they're using fermentation, and I'll get to that later, but in fermentation we have the same organic molecule acting as both the electron donor and acceptor. Oxidative phosphorylation is synthesis of ATP from ADP catalyzed by the protein ATP synthase, which is shown here in pink. Okay, so that entire protein is ATP synthase, and a very important protein found in all cells, very ancient protein, very interesting protein in fact. ATP synthase is energized by the proton motive force. So we see a proton motive force in this figure. That would be a concentration of protons on one side of the membrane generating an electrochemical gradient, both a charge gradient or electrical potential, and also a chemical gradient in that you have more proton chemical species on one side of the membrane than another. Oxidative phosphorylation is the major means whereby all respiratory organisms make ATP. Whether they are conducting aerobic or anaerobic respiration doesn't matter. In both cases, it is the major means whereby all respiratory organisms make ATP. We'll talk a little bit more about this protein in a later part of this lecture. In photosynthetic organisms, ATP is generated via photophosphorylation. Shown here is a diagram of the photosystems. Okay, there's one photosystem, photosystem 2, and then photosystem 3 over here. So these are photosystems, and these are photosystems. They're named in the order of discovery, not in the order in which electrons flow or events happen biochemically. But shown here is a diagram of the photosystems, which are protein and pigment complexes that include many electron-carrying components embedded in a membrane. So each of those photosystems are the locations of pigments such as chlorophyll and accessory pigments, and they are complexed in reaction centers with a variety of highly organized proteins embedded in a membrane. In a eukaryotic phototroph, that membrane would be the thylakoid membrane within a chloroplast, and in a bacterial phototroph, that membrane would be the cytoplasmic membrane. But in both cases, what happens is electrons are transferred from water is the electron donating substance, through the photosystems and two electron transport chains, ultimately to NADP+, the oxidized form, which is then reduced to form NADPH, and it's going to go off to the Calvin cycle. Calvin-Benson is basically just means the Calvin cycle. And what's going to happen there is electrons are going to go to CO2, which is going to result in the formation of sugars. Right? That's carbon fixation. So electrons from water are flowing through photosystem 2, through this electron transport chain, all the way to photosystem 1, through another very short exiting electron transport chain to NADP+, which then reduces it to form NADPH, and then it goes off, and via the Calvin cycle, it's going to be those electrons are going to be attached or associated with carbon dioxide, resulting in the formation of sugars. So that CO2 is the electron acceptor, the terminal electron acceptor, whereas water in this figure was the electron donor. Okay, and that's a photosystem present, and along the way, ATP is generated. Okay, so that electron transport chain is generating, right, a proton motive force. Note the concentration of protons that is greater on one side of the membrane than the other, and note the presence of our friend ATP synthase. That proton motive force is being harnessed here. The protons are flowing through ATP synthase, and that's giving that ATP synthase energy to make ATP. Because light is involved in this process, it's referred to as photo-phosphorylation and not oxidative phosphorylation. The energy source in this case is light and not the oxidation of high energy compounds. And that concludes our discussion for this first of a 4-part lecture on metabolism. Please move on to the next lecture.